What if a plant could breed true? No need for sex, or even a partner. Just seeds containing an embryo identical to the mother plant. This strange and fascinating phenomenon, called apomixis, occurs in numerous flowering plants, a natural form of cloning that agriculture would benefit from using. Why not? Because apomixis has long been a mystery. Today, we have a better understanding of the phenomenon, thanks in particular to the knowledge gained by the Mechanisms of Apomictic Development project. The MAD project, an international consortium bringing together researchers and students from 13 different institutions in Europe, Australia, Mexico, Argentina, and United States of America. During the past 5 years, their findings were communicated through 22 scientific articles. MAD scientists also shared with the public the astonishing diversity of reproductive strategies found in plants through workshops open to students and young researchers, 2 international conferences on apomixis, and several public outreach events in Europe and Argentina. Most plants reproduce through sexual reproduction, where pollen meets ovule and male and female gametes fuse. The 2 parents therefore contribute to the new generation that forms within a seed. With apomixis, no gamete fusion nor male contribution, the seed contains 1 or more maternal clones. We have all come across this peculiar reproductive behaviour. Without even realising it, dandelions and buttercup along roadside and in meadows, many gramineae in grasslands, or even popular fruits such as oranges and mandarins. But no major crop reproduces through apomixis, even though its transfer or engineering could revolutionise agriculture and plant breeding.
This mechanism ensures genetic stability, particularly for high-performing and hybrid varieties. It is therefore a promising tool for developing countries where farmers could benefit from genetic process without having to purchase new hybrid seeds each planting season. The mechanisms that control it are still poorly understood. It is not enough to identify a gene. One must understand how it is expressed when, in which tissue, and how to reproduce this process. It is precisely to meet these challenges that scientists from around the world have come together with a common goal: to unlock the mysteries of apomixis.
To achieve its goal, the MAD project was structured around 4 work packages, the main results of which are presented here. More specifically, these concern the organisation of apomictic plant genomes, the characterisation of candidate genes, and the role of hormones and ovule morphogenesis in the emergence of apomictic reproduction. To decipher the structural and functional characteristics of the region of the genome responsible for apomixis in apomictic grasses of the genera Eragrostis and Paspalum, the researchers will continue identifying the genes in this region, assess their conservation across different apomictic species, and propose hypotheses about their evolution. They also aim to provide a comprehensive understanding of how apomictic genomes function.
By combining long-read sequencing technologies with high-throughput short-read methods, we had fully characterized the genome of Paspalum notatum and identified nearly 45,000 genes models. In the apomictic plant Q4117, we identified the aposporic controlling locus, a 30 megabase pair genome segment containing approximately 2,500 genes. In parallel, we have functional characterized 2 apomictic candidate genes named Qui-Gon gene at TGS1 during the reproductive development and confirmed their involvement in aposporo embryo sac formation and parthenogenesis.
Similarly, they have obtained and characterized the genomic sequence of other tropical apomictic forage grasses, such as Eragrostis curvula. Here again, comparisons with the sexual diploid ascension revealed a single region that they characterized for its genetic content. With this new knowledge, their current work is focused on deciphering the role and evolution of these specific regions. Apomixis is a complex process where thousands of gene products and other molecules interact in the ovule. In the second work package of the project, researchers have focused on key genes that control the molecular cascade, leading to the transition from sexuality to apomixis.
The activity of a gene can be detected through a molecule it produces, called RNA. In our case, we studied species like Paspalum notatum, Eragrostis curvula. Some reproduce sexually, while others use apomixis. So we extracted total RNA from ovules at different stages of development and sequenced it. Then we compared the list of genes that were active in each case. This allowed us to find genes that were consistently more active or less active during apomixis. Interestingly, some of these genes are located in a specific genomic region that controls apomixis.
Once identified, the next step is to validate experimentally whether candidate genes truly control the trait.
We create plants where the gene is either turned off or alternatively where the gene is artificially activated. Then we observe what happens. This is the kind of experiment we used to identify genes like TGS1, which works as a repressor of apospory, the first step in apomictic reproduction.
The third work packet is devoted to the study of hormones that are pivotal signaling molecules participating in the regulation of numerous processes in living organisms, including reproduction. Here, the researchers asked whether these hormones actually direct apomictic reproduction.
One of the first clues we found was that many of the genes show an unusual activity in the ovules of apomictic plants related to hormone pathways, specifically the production, transport, and response to hormones like auxin, such as monic acid, and cytokinins. For example, when we artificially activated a gene called R10, which responds to auxin in the ovule of a sexual plant, the plant began to form multiple unreduced embryo sacs, which is a characteristic featured by the apomictic pathway.
But is there a direct way to test whether hormone levels differ between these two types of plants?
Yes, we can measure hormone levels in both sexual and apomictic plants using chemical analysis. But during the MAD project, we introduced a special plasmid, a small circular piece of DNA into both sexual and apomictic individuals. This plasmid carried a gene that produces a fluorescent protein, but only in cells where auxin levels are high. These genetically modified plants are already growing, and now we're just waiting for them to flower, so we can observe, compare the hormone signals during seed development.
The objective of the fourth work package of the MAD project is to understand how the architecture and shape of the plant ovule determine changes in germinal cell identities in apomictic plants. We wish to precisely describe how ovule cells gradually arrange and communicate with each other in order to define which of them will enter the germinal program to form a seed.
To achieve this, we studied ovules in 3 dimensions, because plant ovules are small domes rather than flat organs, and understanding their structure requires 3D observation. We use specific microscopy techniques combined with computational tools to reconstruct digital ovules in 3D at every developmental stage, capturing every single cell. Using these models, we measure the size and shape of cells, as well as their spatial arrangement, to infer cell-to-cell communication. We then compare germinal cells with their surrounding somatic cells, and further compare sexual and apomictic plants, identifying differences not only in germinal cells, but in the overall architecture of the ovule, the arrangement of cells, their size, growth timing, and dynamic changes of dome shape. Our main findings is that ovule morphogenesis conditions the plasticity of cell identity and the fate of germinal cells, both in sexual and apomictic plant species.
Researchers involved in the MAD project and other initiatives around the world are making progress in unravelling the mysteries of apomixis, making seeds capable of faithfully copying themselves closer to the fields every day. In the face of climate change and growing food demand, understanding asexual reproductive behaviours opens up new opportunities for managing biodiversity in natural clonal populations and delivering the promise of more sustainable and equitable agriculture.